Okay, we're live. Yeah. Welcome, everybody. I'm sure everyone's slowly streaming in now. So welcome to the third CSEE PhD Excellence in PhD Research Award talk. First two have been really incredible, and we're really excited to bring in another one of our winners, Sarah Amundrud from UBC, who is going to be talking about drivers of species distributions and ecological communities. I'm going to let her introduce herself as well and start sharing her screen and started her talk. But first, I just wanted to say, like the previous sessions, if you've managed to tune in for them, just like those sessions, we'll have a question period after, a live question period with Sarah afterwards. And so you'll see on the YouTube, there's a chat bar on the side. And so if you have questions at any point during the talk, whether before, whether during or right after as well, you can post those questions in the chat and I will ask them to Sarah afterwards and you'll be able to get an answer. So it should run fairly smoothly so far. It's been a pretty great experience, and we're, like I said, really excited to have Sarah here to talk about drivers of species distributions and ecological communities. So without further ado, I'm going to let Sarah take over. Yeah.
All right, well, thanks so much, Peter, and thanks to CSEE for the award and for this opportunity. So today I will be talking about some of my PhD research at the University of British Columbia, and most of my work focused on studying the drivers of species distributions and ecological communities across spatial scales. So I was particularly interested in exploring how the interplay of abiotic and biotic drivers is affected by changing environmental conditions, such as with climate change. So before I get started, I would like to quickly acknowledge my supervisor, Diane Sherivastava, and my co-authors. Without them, my research would not have been possible, as well as all the funders and the various institutions and organizations in Costa Rica that enabled this research. So for my primary research for my PhD has been investigating the drivers of species distributions and by extension, the makeup of ecological communities. So species distributions in space are linked to species ecological niches, and Sober-Ron differentiated between two types of niches. So the Grinnellian niche, it is defined by environmental conditions, and the Eltonian niche, which is defined by biotic interactions. So both types of niches are relevant to understanding the distribution of species. However, the Eltonian niche is easier to measure at fine scales that are characteristic of most ecological studies, while the Grinnellian niche is better suited to low spatial resolutions at which we describe species distributions. So climate change can affect both. First, climate change obviously directly alters environmental conditions, such as temperature and precipitation, but climate change can also affect species interactions, such as by altering feeding rates and thus interaction strength of species. So I was interested in whether climate change can affect the relative importance of environmental processes and biotic interactions, and whether these effects depend on spatial scale. So I did my PhD work in Costa Rica, where climate change is not only expected to increase temperatures, but it's also expected to decrease precipitation. So the map you can see here shows the expected change in precipitation by the end of the 21st century. And it shows that in Costa Rica, precipitation is expected to decrease by about 30%. And that, of course, will lead to increased frequencies of droughts. Now, in the neotropics, bromeliad plants collect rainwaters between their leaves, as well as detritus, which are just dead leaves that fall from the trees and that collect between the leaves of bromeliads. So these bromeliads provide habitats for aquatic invertebrate food webs, and these food webs tend to be quite diverse. So they often contain several functional feeding groups and trophic groups, and we know that species interactions are very important in the system. For example, the damselfly larvae that you see on the very top here is a keystone predator, and this damselfly is really important in structuring the community. So bromeliads make really great model ecosystems because they are natural systems with complex and realistic food webs. So there's this element of realism. They're also easily manipulated. So we can easily add species or remove entire species from the system. And there's also many of them. So we have replication, which we don't typically have for a lot of other ecosystems. So for my PhD, I used a variety of research methods to investigate the drivers that shape bromeliad food webs and insect distributions across a range of spatial scales. So at the large spatial scale, I used species distribution models, and I did this to study the effects of climate and competition on the damselfly distribution. Then at the landscape scale, I used observational surveys along environmental gradients to study insect distributions along elevation. And then at the local scale, I used manipulative experiments to study the drivers of community composition. So for today, I will focus on the research I did at the regional and at the local scale, just because I won't have time to talk about everything. So I'm going to start with the study I did on the regional or landscape scale. So to study the drivers of insect elevational distributions, I conducted an observational survey of bromeliad food webs along altitudinal gradients in Monteverde, which is in northwestern Costa Rica. So at Monteverde, there are two elevation gradients at which I conducted my study. So the Pacific slope is on the left and the Atlantic slope I'm showing on the right. And both slopes exhibit a gradient in temperature. So it's pretty cool on top, but if you go lower in elevation, it becomes warmer. But there's the two slopes differ in their humidity. So the Atlantic slope has very high humidity, while the Pacific slope is relatively dry and experiences frequent droughts. So another important difference between those two slopes is that the damselfly, which is that important keystone predator I told you about earlier, is only present at low elevations on the Atlantic side. So this is consistent with the species distribution models I did for damselflies, which suggested that this predator is limited by cold and by dry conditions. So I was interested in what factors determine the elevational distributions of bromeliad-dwelling insects. And in particular, I was interested in the relative importance of temperature and biotic interactions. Okay, so I was really fortunate in that I was able to take two research teams with me down to Costa Rica, who helped me with this research over 2 years, 2015 and 2016. And in order to conduct the survey, we worked in two private reserves. And a lot of the area there was only accessible by foot. So we did a lot of hiking and actually a lot of multi-day field trips to really remote research stations, which was really, really cool. Bromeliad sampling involved just collecting bromeliads, removing them from the trees, then rinsing out the entire contents and sorting through the contents, and then identifying and counting all invertebrates that were in bromeliads. So in order to get an idea whether different insect species have different heat tolerances, we experimentally determined their critical thermal maxima. And we did this by placing individual insect larvae in water baths that we then slowly heated up at a constant rate. And we observed the insect. Once it stopped moving or responding to a tactile stimulus, we then recorded the temperature as the critical thermal maximum. So that was our index of heat tolerance. We then investigated whether heat tolerance can explain the elevational distributions of insects. So the thermal adaptation hypothesis predicts that species with a high heat tolerance would occur at lower elevations where it is warmer. And that's because they can withstand higher temperatures. So insects would be adapted to the temperatures to where they live. So what we found is that on the moist Atlantic side, the results agreed with the thermal adaptation hypothesis. So on the x-axis here, you can see heat tolerance. So insects towards the right are more tolerant to heat. And on the y-axis, you see the mean elevation of occurrence for each species. So on the Atlantic side, temperature can explain the elevational abundance patterns of insects. Next, we looked at the Pacific side. So when we looked at that side, we found no convincing evidence that heat tolerance varied with elevational distribution patterns. And there's several potential reasons why that could be. So first, we are aware that there's extremely low power, so we only have 4 species. And also the species that was driving that pattern on the Atlantic slope, which is the damselfly, was entirely missing on the Pacific side. But maybe there's something else that's actually more important in explaining this pattern. So in order to explore other potential drivers, we employed structural equation models, and that was to investigate whether species interactions might be better at explaining those elevational distributions. So we were particularly interested in whether interactions from potential predators are important. So on the Pacific side, we included tipulates as potential predators. So tipulates are crane flies. So these are the larvae of crane flies, which sometimes in other systems can act as predators. And then we performed 3 separate structural equation models. So basically 1 model for each of the remaining focus species. So what we found is that on the Pacific slope, negative effects of tipulates mediated the effects of elevation on 2 of the 3 focus species. So species interactions seem to be important on the Pacific side. On the Atlantic side, we included tipulates and damselflies as potential mediators. So again, the 2 potential predators. But there we actually found no evidence that any of those species mediated the effects of elevation on the abundance or on the distribution patterns. So in summary, with this study, we found that the relative importance of temperature and species interactions in explaining those elevational distributions of insects depended on mountainside. So when conditions were wet on the Atlantic slope, heat tolerance was important. But when conditions were dry on the Pacific slope, species interactions were more important. So that then made us wonder why tipulates or crane flies had such strong negative effects on other insects and why that only happened on the Pacific slope. And that now brings me to the next part of my talk. So now I'm moving down to the local scale. So first I'm going to tell you about some experiments that we did to study the effects of drought on the trophic role of tipulates. All right, so bromelia tipulate larvae usually are considered to be important detritivores. But when I started working with them, and I've also done my master's in the same lab, so I've been working with them for a while now, I sometimes observed them consuming other insects. And so I started to wonder if this predatory behavior in tipulates is somehow related to drought. So for my PhD, I decided to do an experiment that tested first whether tipulates become predatory under drought conditions, and then if yes, what the mechanism is. So why would they become predatory? So my assistants and I performed 2 microcosms experiments. So for the first experiment, we performed a factorial manipulation of drought and tipulates. So for drought, we actually had 2 water treatments. So we either had water present or water entirely absent. And then for tipulates, we had tipulates present, absent, and then we also had a caged tipulate. And that was in order to differentiate between any consumptive and potentially non-consumptive effects the tipulates have on the community. So in this graph here, we have the 6 types of treatments on the x-axis. So these are all the possible combinations of drought and tipulate treatment. And the y-axis shows the number of prey that survived until the end of the experiment. Okay, so first we found that drought had a direct negative effect on the prey community, which was not very surprising. Then there were also some interesting non-consumptive effects. So we found that non-consumptive effects arose when water was present, but not when water was absent. But the really interesting result was that tipulates had no consumptive effects on other insects when water was present, but significantly reduced prey survival when there was drought. So this confirmed our hypothesis that tipulates do become predatory under drought conditions. So then, of course, we started to wonder, okay, what's causing this? Like, what's the mechanisms of tipulates becoming predatory under drought? And we came up with two potential hypotheses or ideas. So the first hypothesis we had is that desiccation stress changes the nutritional needs of tipulates, that drought somehow makes them require more protein and thus switch to insect food. And the second hypothesis was that drought increased random encounter rates of tipulates with prey, because you can imagine the removal of the water column would compress the habitat, in this case from a 3D environment to a 2D environment, and that then would increase encounter rates. So we tested these hypotheses by performing a second microcosm experiment. It was pretty similar to the first one, but instead of removing the water entirely for the drought treatment, we just reduced the water level. And that was in order to not impose a desiccation stress. So if desiccation stress causes tipulates to become predatory, we would expect negative results of this experiment because tipulates were not subjected to a desiccation stress. If, however, increased encounter rates due to habitat compression caused tipulates to become predatory, we would expect tipulates to become predatory in this experiment as well as the first experiment. And that is because, as I mentioned before, if we shrink the water level, that would then compress the water column and the habitat and lead to increased encounter rates. So the x-axis on this graph shows the duration of the experiment, and the y-axis shows prey survival. And what we found is that when tipulates were absent, so these two lines show communities without tipulates, then prey survival slightly decreased over time, but there was no difference between whether water level was high or water level was low. So this is just some background mortality that's happening anyway. So next I'm going to show you communities where tipulates were present. So when the water level was high, tipulates again had no effect on prey survival. So it's pretty similar to what the background mortality is. However, when the water level was reduced, tipulates significantly decreased prey survival. So this result supports the second prediction and is suggesting that tipulates become predatory under drought because reduced water level increases random predator-prey encounter rates. And maybe this has some important implications for other ecosystems that are prone to dry out as well, such as ponds. So just a quick recap of what we have learned so far. So on the scale of the landscape, we found that the relative importance of temperature and species interactions in driving elevational distributions of insects depended on environmental conditions. So specifically, when conditions were dry, species interactions became more important than abiotic effects. And this is because drought enabled tipulates to become predatory. So this has really important implications for this ecosystem, given that climate change is expected to increase the frequency of drought. However, drought is not the only variable that will shift this climate change. So one of the difficult aspects about predicting the effects of climate change is that multiple climatic variables shift simultaneously. Then there's also uncertainty about whether or not species can adapt to changing environmental conditions or track climate change by migrating. And that now brings me to the last part of my talk, where I combined multiple experimental approaches to see if we can understand some of these multifaceted effects of climate change. All right, so in this study, I performed another microcosm experiment where I manipulated climatic stressors, prior exposure of species to future climatic conditions, So this is to take account adaptation or migration, so tracking climate change, or and species interactions. So bromeliad communities on the Pacific slope of Monteverde actually presented a perfect opportunity to conduct such an experiment. So at the top of the mountain, current conditions are relatively cold and wet. So I use this as my focal area representing the current climate. And then predictions for climate change are rising temperatures and a rising cloud bank. And that is making conditions more similar to what they are currently at the lower elevations. So in order to manipulate warming, I used a space for time approach, where I transplanted high elevation communities to the site called Monteverde, the high elevation site, and that is to mimic the current climate, as well as to the site UGA, which is the site that mimics the future climate. So as a second climatic variable, I manipulated drought length in microcosms, and I had drought length treatments that ranged anywhere from 0 to 22 days with 2-day intervals. So I had everything from no drought to very severe droughts and everything in between. In order to manipulate prior exposure of species to future climatic conditions, I used a common garden approach, in which I transplanted communities that were collected at low elevations and thus already exposed to those future climatic conditions to the UGA site. And then finally, to manipulate species interactions, I added tipulates to half of the communities, because we already know that they may have important effects on the food web. So that then allowed me to see whether tipulates mediated any indirect effects of drought or temperature on the insect communities. Okay, so to summarize the experimental design, because it was pretty complicated, so the common garden is comparing the red and the green communities to each other. So the green ones are not adapted to the future climatic conditions, but the red ones are potentially adapted. And then the space for time experiment compared the high elevation communities that represent the current climatic conditions to the low elevation communities representing the future climatic conditions. Okay, so this graph here is showing the treatment effects on prey survival. So we have drought length on the x-axis, and on the y-axis we have prey survival as the response. And the 3 types of communities are indicated by color. And then the tipulate treatment is indicated by line style. So the dashed line shows the treatments or the communities with no tipulates, and the solid lines show the communities that had tipulates added. So there's a lot of information in this single figure, but the first thing, or one of the first things you probably noticed, is that prey survival decreased as drought length increased in all communities. So there are negative slopes for all of those communities. So there was a negative direct effect of drought. Then we also found that the effects of drought were stronger under warm conditions. So that came out as a significant interaction of drought with the space for time treatment. And to illustrate this, I'm showing you here the partial residuals for the space for time treatment. So the high elevation communities are shown in blue, and the low elevation communities are shown in orange. And as you can see, warming intensified the negative effects of drought. So there was this significant interaction. There's a steeper slope in the warm communities. So this result then suggests that the multiple climatic stressors can interact, and that of course can complicate predictions we can make for climate change and the ecological effects. So when we looked at the common garden treatment, we actually did not find any effect whether species had prior exposure to future climatic conditions or not. So I'm not going to go into this anymore. So next we examined whether tipulates mediated the effects of simulated climate change. So first, we found that tipulates negatively affected the prey community under drought conditions. So again, that's consistent with the first 2 studies I talked about before. So that came out as a significant interaction of drought with tipulate treatment. And these figures here again are showing the partial residuals for the tipulate presence versus tipulate absence treatments. So the three types of communities are actually shown on separate plots here, indicated by the different colors again. So when tipulates were present, which are the dashed lines in those plots, then drought only had minor effects on the prey community. However, when tipulates were, sorry, that was when tipulates were absent, the dashed line. But when tipulates were present, which are the solid lines here, then drought had strong negative effects on the prey community. And that was consistent across all three communities. So these results now suggest that tipulates mediated the effect of drought on this community. And that's an example of where the indirect effects of climate change that happen because of altered species interactions are actually more important than the direct effects of drought alone. Okay, and finally then, we looked at the combined effects of warming and tipulates. And here we found that warming intensified the negative effects of tipulates. So again, this was apparent through a significant interaction of tipulates with the space for time treatment. And to illustrate this, I'm first showing you the effects of the space for time treatment when tipulates were absent. So the communities subjected to the current climate, shown in blue, and the communities subjected to the future climate, as shown in orange. And as you can see, the space for time treatment had no real effect on prey survival when tipulates were absent. So these lines pretty much overlap. Now let's have a look at what happened when tipulates were present. So in the presence of tipulates, communities at the warm location exhibited significantly lower survival than communities at the cold location. And that was especially true for long droughts. So again, we only see an effect of warming when tipulates were present. And this is again suggesting that indirect effects of warming were actually more important in the system than direct effects of warming, because we didn't really see a direct effect of warming at all when tipulates were absent. Okay, so this experiment demonstrated how complexities arise from the interplay of biotic and abiotic mechanisms of climate change. And I think the most important take-home message is that indirect effects of climate change can sometimes be much, much more important than the direct effects of climate change from just change in temperatures or precipitation. So here, tipulates mediated those indirect effects. So this research is actually part of a growing body of evidence that is suggesting that single species can drive the ecological outcomes of climate change. So Chris Hardy, who is also at UBC, used the term leverage species to describe species for which climatic impacts result in those sweeping community level changes. However, it's still really difficult to predict a priori what species can actually act as leverage species. So hopefully, as we are collecting more examples like this one, we can find generalities that help us better predict which species will likely act as leverage species. So I'd like to end my talk with a fun fact. So it actually turns out that I couldn't have given this talk on a more appropriate day. And maybe some of you have heard, but today, May 15, is World Crane Fly Day. Yeah, so happy World Crane Fly Day, everyone. And with that, I'd like to thank everyone who saw my talk. I know it's the beginning of a long weekend, so thanks for coming anyway. And I'm happy to take any questions.
Awesome. Thank you so much, Sarah. That was an amazing talk, amazing pictures. I'm so jealous of your work.
Thank you.
And yeah, terrific job. For everyone tuning in, if you have questions, you know, pop them in the chat right now below the YouTube stream, and I'll ask them to Sarah. I actually had a question for you. I was wondering, in the beginning portion of your talk, when you were talking about the effects of or kind of these abiotic factors, and you checked the kind of the critical thermal limit of species. I was wondering whether you checked other kind of critical limits as well, things like precipitation, for example. Was that significant, or how did that look like?
Yeah, that's a really good question. So actually we also wanted to look at drought tolerance and whether differences in drought tolerance can explain those elevational distributions, especially in the Pacific side. And we did an experiment, and unfortunately it just didn't work out. So it was this massive experiment. It was a ton of work, but yeah, it just didn't work. So we can't really make any conclusions on it. But that saying, so tipulates actually, they seem to be quite tolerant to drought. So during my master's, I did an experiment there. I measured drought tolerances of 10 bromeliad insect species, but that was at a different study site. But that suggested that tipulates are quite tolerant to drought. And that might actually explain why they do so well at the low elevation Pacific side where it is so dry.
Okay, wow. Yeah. Thank you for that answer. A lot of applause streaming in as well. People applause, clapping hands. Very nice talk. Thank you. And a question here. Someone's wondering, how would you explain that the community from cold, wet environments is more resilient to drought, less prey dying, compared to the communities from the warm, dry environments? Wouldn't you expect the opposite, considering communities would be habituated to their environment?
Yeah. And just go back to the slide. So the question was like, why are communities at the cold environments more resilient? Oh, okay. Yeah. So that probably had to do with the location. So so like the way that we manipulated drought is we assembled communities in microcosms. We left them there for a day. Just to get used to the microcosms, and then we removed the water column from for all the drought treatments. So the communities now that were at the high elevation cloud forest, it's actually quite moist in there. So it took the microcosms a lot longer to dry out than at the lower elevations where it was quite dry. And that actually would explain why the high elevation communities responded a lot slower to drought. So I hope that answers your questions. I'm not sure if I understood it correctly. But...
Yeah, okay, thank you. Another question coming in. Hello, cool job, cool field site. I'd echo that again. Cool job, cool field site.
Yeah.
They're wondering, for the first part about the CT max measurement, how many individuals were you measuring per species? And did you investigate whether species have different individual variations in CT max that might be related to altitude?
So we measured a lot of species. So we had a total of 9 sites along both those 2 elevation gradients, and we did 10 individuals per species per site. So it was like 90 per species, as long as they were present. So not all species were present at all sites, but when they were present, we did 10 individuals. We did this for 5 species. And sorry, what was the second part of the question?
And whether, do you investigate whether species have different individual variations in CT max and whether that might be related to altitude?
Yeah, so we in another study, or like another analysis that I didn't show today, but it's like we're actually just working on a paper right now, where we looked at how within species variation changes along those elevation gradients, and it does look like there is some adaptation. So as you within one given species, as you go down in elevation, their CT max actually tends to increase. However, it doesn't really keep up with the actual temperature increase. So now if you would look at the temperature of the habitat versus the CT max, that gap becomes narrower as you go down elevations. So low elevation species are actually more vulnerable to warming.
Wow, interesting. Yeah. Is another one coming in, and the previous question asker said, that makes sense, thank you. And another question, You mentioned that you didn't see any effects of drought or interactions in your common garden. What do you think is going on with that?
Yeah, that's a really good question. And I'm not sure. So maybe there is enough dispersal between those two sites. So even though those sites, there was a really big elevational gap. So it was about 500 or 600 meters in elevation. So the temperature difference there was actually about 4 to 4.5 degrees. But these are flying insects when they are grown up. So there could be some gene flow, and actually we don't really see that effect of adaptation. And we did look at adaptation, as I just said, to temperature. And actually on this specific site, between those two sites that I mentioned, for most of the species, we didn't really see a significant difference in CT max. So it didn't really look like most of the insects were in fact adapted to the local temperature. The only species was the tipulate, where there really was an effect of the lower elevation and the high elevation on their temperature tolerance. So this is the only species that might actually be adapted to those conditions.
Do you think that it's a predator species makes a difference?
I'm not sure. Yeah, I don't know. Yeah, good question. I don't know. Maybe, well, typically, I guess predators have longer generation times, and therefore that might have something to do with it, but I'm not actually sure if that's even true for tipulates. Their generation times really aren't that much longer. So in this system, it's really the damselfly who have those really long generation times, and they weren't present at all at the Pacific side.
Okay. I'm curious as well, you mentioned a bit about the dispersal. How far do they disperse as adults? Do they travel from one side, from the dry side to the wet side, for example, of the mountains as well?
Again, I don't think we know at the moment. So so when I was in Monteverde, I actually collected a lot of insects to look at this. So we sent those samples to collaborators at Rutgers University, and they were going to look at gene flow between those sites. And we're actually still waiting for the results on this also. So like we have some interesting results regarding what species are actually there, and there's a lot more variation than we thought from just looking at it morphologically. But I don't think we know much yet about the dispersal.
Awesome.
But
Thank
yeah,
you.
hopefully we will soon.
Yeah, it's a massive project. It's so cool. There's so many different avenues off of it.
Yeah, thank you.
Yeah. One more question from someone you might know, Diane, actually. Do you think that these results can be extended over geographic space to other locations, or are they Monteverde specific?
I think it would be really interesting to do this at other locations as well. Like I think those tipulates are really common, not just in Costa Rica, but they are one of those species that really occur at most places where we find bromeliads. So I actually, yes, I do think that it probably would hold up in other locations as well.
Awesome. Do you think there's often, I guess there's a difference between kind of tropical and temperate species, kind of like a division there, especially since tropical species are generally operating near their upper thermal limit for more parts of the year. Do you think that might influence how much it can be extended over?
I'm not sure actually. I'd really have to think about that. Yeah. So yeah, I don't know.
No, that's good. Yeah. Awesome. If anyone has some final questions, I guess we'll put in one or two more if people have something else. I guess I'm curious, you had one in your kind of breakdown, you had like a final bullet point at the top with, I think, species distribution models. I'm just curious if you could quickly, maybe in like a few sentences, break down how that related to it as well.
Yeah. So like we know that the damselfly, that keystone predator I told you about, is quite important in the system. So this was actually a project that was started by a former postdoc in the lab who collected all the data, but then they never got around to doing the model. So that's where I came in. And so for this, we were interested in and what are the drivers of the geographic distributions of the species? So is it climate, or is it potentially physical barriers to dispersal? So they didn't really seem to be south of the Andes mountain range, for example, and they also weren't on the Caribbean islands, so they could be this effect of the ocean and mountain barrier, or could it be competition from other damselflies that also spend their larval stages inside bromeliads? So we don't really see a lot of like other damselfly species in Costa Rica or in Central America, but they're quite common south of the Andes. And then what we found is that really physical barriers to dispersal were really important, and climate as well. So specifically, they seem to be limited by cold temperatures and by dry conditions, which is totally what we also found then at Monteverde Mountain. So that was quite consistent. And then competition really didn't come out in the model at all. So it seems like at those large spatial scales, I don't want to say they're not important, but we can't really detect them at such a high, low resolution at those large spatial scales.
Okay. Wow, fascinating. Thank you so much. We'll have to get you back sometime to give the talk on that other chapter as well.
Okay.
And thank you for a terrific talk. Congrats again on winning our CSEE PhD research excellence in PhD.
Yeah, thank
research
you so
award.
much, and also thank you so much for organizing all those talks. I'm sure it's a ton of work, so yeah, I'm sure we all appreciate it very much.
Thank you. It's a pleasure. And thank you on that note for everyone tuning in and who asked excellent questions. We have one final one, if you don't mind, Sarah. Do you mind if
I thought it was off the hook now.
Sorry. One final one. Did you see overall effects of climate change on biomass of these communities, either through direct or indirect effects?
I never looked at biomass, so I have abundance. I wasn't like, we did do some measurements of like insect lengths, but like we were at those remote stations, we didn't have electricity or anything, and we went through a few hundred of individuals per bromeliad, so we just weren't able to actually do biomass. So I could tell you about abundance. I have the data for abundance. I actually don't know it right now, but the person who asked a question, feel free to email me about it and I look into it for you.
Awesome. Thank you so much. Now you're officially off the hook. Thank you again for an excellent talk and for taking a bonus question there. Thank you everyone for tuning in, and hopefully we'll see you all next week for our next